Hello, my name is Camille Condron, an undergraduate student from Dr. Chang's lab, and today I'll be presenting to you the effect of drought-tolerant bradyrhizobium biofertilizer on soybean growth in a temperature gradient greenhouse. The purpose of this experiment is to test biologically relevant temperature change and their effects on soybean growth. The temperatures are predicted to go up by 2 to 4 degrees Celsius in the future, which will vastly affect agriculture. This is shown by the graphic in the bottom left-hand side. In this study, we tested the effects of temperature stress, which is a different type of stressor compared to drought stress, on our inoculant via a novel temperature gradient greenhouse to test the effects in normal temperature, 2 degrees Celsius increase, and 4 degrees Celsius increase. It is important to note that drought stress relates specifically to the ability in which a plant can maintain its biomass production during arid or drought conditions, while temperature stress is more related to the rise in temperature beyond a threshold level for a period of time. Due to that increase in temperature, there is sufficient irreversible damage to plant growth and development. Temperature stress is a growing issue that can cause up to 25% yield loss. In 30 years, it is anticipated that we will see a temperature increase of 4 degrees Celsius, which is why we wanted to see if we could gauge the success of our inoculant under those imposing conditions. Moving on to our methods, we had 3 treatments. We had our Texas VA drought-tolerant inoculant, our control group, and CellTech, which were tested in a novel temperature gradient greenhouse in order to test the effects of temperature stress. How this was done was by building a greenhouse with a filtered fan unit and that had mesh put around it on one end of the greenhouse that takes the outside air and pulls it in and then pushes it throughout the greenhouse. The plexiglass dividers, which are represented by the black bars in the graphic towards the upper middle, represent the dividers that were between each of the temperature sections. The darker gray areas represent where the replicants were planted. The air is heated in each section before moving through the holes in the plexiglass dividers to the next section. There's an even heating of each of those segments. None of the plants experienced any drought as they were all watered equally and regularly via drip hoses. Another aspect of the methods I would like to mention would also be the medium of what the plants were grown in. The soil was mixed of 2/3 local topsoil and 1/3 potting mix to allow drainage with only 1/6 the fertilizer amount as to not inhibit nodulation. Each seed has 1 milliliter of fertilizer, usually way less, but we were testing for effects of our overall combinations. There were a total of 18 plants per temperature gradient section, and in each of those sections there were 6 replicates of each treatment group. Half were taken at mid-harvest, and the second half for final harvest. As you can see in one of the photos in the middle, 3 replicates were taken, and those are what their root systems look like. Root systems of these plants are very tedious to count. Moving on to our results, the data that was collected at mid-harvest and final harvest were our height and biomass measurements. Height at mid-harvest and final harvest of the inoculant indicated increases in the ambient 2 degrees Celsius and ambient 4 degrees Celsius, which is leading over CellTech. At the final harvest, plant height, plant weight, and main stem pod number measurements all indicated an increase in the ambient plus 2 degrees Celsius. Not much of an increase in the ambient 4+ degrees Celsius increase. It is important to note that only 1 replicate from the Texas VA 4 degrees Celsius condition died, which may have skewed the results a little bit. In conclusion, based on those results from the 9-week harvest variability and the total pod weights and main stem pod number of our inoculant and ambient 2 degrees Celsius plus data, we have shown to improve the growth of the plants long-term under air temperature stress. If we were to make any future edits to the project, which we probably will, we would need to test a drought-sensitive cultivar. TN16 is really too new to be deemed drought-tolerant, but its parent cultivar is drought-tolerant, which is why we used it initially. This is one thing we would change with our experiment. It also would explain why the data trend for the control and why it doesn't move as much as you can see in those orange bars. They stay kind of consistent throughout everything. So that would explain why there is not much of a trend there as we were anticipating. We would also do a DNA extraction to perform bulk and rhizosphere soil microbiome analysis in addition to completing the nodule counts and dry weight analysis. Thank you so much.